We have just one more thing to say about the control of hormones, and it has to do again with your target cells. The target cells being the downstream receivers of signal, where receptors should be your key word that you're thinking about there. Think about all the ways that receptors are manufactured and so on. So when we talk about receptors, there's two different ways that we have receptors, and this helps us to classify our hormones into two different classes. So let me just show you these and toggle back and forth just a second. There are two major classes of hormones. There are amino acid-based hormones, which are shown here. There they are. And there are steroid hormones, which are shown right here, there, and there. So those are the two classes of hormones: amino acid-based hormones and steroid hormones. All right, so let's talk about the way that amino acid-based hormones work. Amino acid-based hormones work through a second messenger system. They are polar. They are amino acid-based. Amino acid-based structures are basically polypeptides, or they could be considered small proteins. And as a result, they are considered overall polar entities. Polar entities may not move through the plasma membrane. Here's a plasma membrane. Remember that it's a bilipid membrane, the phospholipids here back to back with each other. The phosphate head sticking out, the lipid tails sticking through the middle. And so the bulk of the plasma membrane is made up of nonpolar lipid tails, and therefore nothing that is polar can actually move directly through them. So all that to say that amino acid hormones must remain outside of the cell. So they are the type of hormones that will utilize those transmembrane receptors. Here it is. There's a receptor. You see it's outside and inside, so it's across the membrane. That's why it's called transmembrane. So these transmembrane receptors have the ability to receive signal from outside of the cell. Here we have our extracellular fluid up above. Our intracellular fluid is down below, which is also called the cytoplasm. And so here's the hormone staying outside of the cell because it cannot enter, because it's polar and it can't make its way through those nonpolar fatty acid tails. Okay, so it interacts with this huge, massive protein, which is called a receptor. The receptor then is going to interact with our G protein. Here's the G protein. Now, this is sometimes called the amino acid action here, is sometimes called a second messenger system, and is sometimes called a G protein mediated system. So let me just actually show you through like what happens at the end. And then I'll go through all the little details. I think that might be a little easier. So we have our hormone outside of here. It interacts with the receptor. At the end of the day, what happens then is that there are massive changes made inside of the cell. In this case, it's the activation of a protein kinase. So that's what we're trying to get to. The problem is that you want this change to happen inside of the cell, but the hormone must remain outside of the cell. So we have this kind of elaborate mechanism that allows this hormone outside of the cell to eventually make this change inside of the cell. Okay, so a receptor receives the hormone. It then interacts with the G protein. The G protein is so called because it utilizes GTP, which is a close cousin of ATP. The GTP then is converted to GDP, and as a result, that is an endergonic reaction. That's an exergonic reaction that allows for the endergonic reaction of this G protein changing its configuration. We have a G protein here in its inactive form. Once it's activated by GTP, you see that it changes its form. It becomes a different shape while the GTP is attached, and it makes its way down the kind of like the inside of the plasma membrane and interacts with this next structure in line, which is called adenylate cyclase. Adenylate cyclase, as the ASE there at the end of the word indicates, this is an enzyme. So adenylate cyclase is an enzyme. It receives that activated GTP, and that's the signal for the adenylate cyclase to start doing what it does. Adenylate cyclase is actually very important and very well known in biology circles because it is the thing that can convert our ATP friend, and I think you know what ATP is. It's the energy currency inside of the cells. It comes about by the metabolism of nutrients such as glucose. So here we have our nice ATP friend. In this case, rather than using ATP for energy, we're going to take our ATP and we're going to convert it into this guy here, CAMP. It actually stands for cyclic AMP. Now, CAMP is very well known as well. It's incredibly important. And as long as the adenylate cyclase is activated, it will do this many times. It will take lots of ATP and convert it into cyclic AMP over and over and over again. The cyclic AMP can then go on to do lots of special things inside of the cell. So this basic concept here, ATP to CAMP, all of this is stereotyped. In other words, it always happens exactly the same way. Receptor, G protein, activation by GTP, adenylate cyclase, and then ATP to cyclic AMP. All of that always happens exactly the same way. The question is, what does cyclic AMP do next? And in this case, it's showing one example of what it can do, which is very powerful. It can take this inactive protein kinase and make an active protein kinase, which then allows for the phosphorylation of different structures inside the cell. And since we're making lots of cyclic AMP, this can happen many times. We can take the cyclic AMP and activate lots of these protein kinases, so you can continue to have tons of these phosphorylation events inside of the cell, which is a very powerful way of energizing certain systems inside of the cell. And yeah, so it's it's what we call a cascading event. It does not take much hormone at this level to cause lots of things to happen at the level of the cyclic AMP. So, for example, one little hormone could allow for a lot, like thousands of activated protein kinases to be present inside of the cell. So that is the amino acid system. Let's take a look at the steroid system. So just so you know, one of the things that you might be asked about, or one of the things you might just kind of want to be considering about, is when you're looking at different hormones, which we'll be discussing down the line, almost all of the hormones are amino acid-based hormones. So almost all hormones utilize this second messenger system, or this G protein coupled mechanism, in order to do what they do. Only a small number of hormones are steroid hormones. And so, in my opinion, the easiest way to remember them is just to memorize all the steroid hormones. There's not that many of them. Most of them are familiar to you. And so then you can just kind of default back to, well, if it's not one of the things on this list, it's probably an amino acid hormone. You know what I'm saying? Okay, so let's talk about a steroid hormone. A steroid hormone is a fatty structure. Steroid hormones are based on cholesterol, which is a fat. And since they are derivations of cholesterol, they are able to, and they are fatty and they are nonpolar, they're able to move right through that plasma membrane. Remember that with the amino acid hormones, they could not do so and must remain outside of the cell. That is not a limitation with steroid hormones. Steroid hormones can pass right through the plasma membrane because nonpolar likes nonpolar, so it moves right through the plasma membrane. And in this case, remember that I said some receptors are buried inside of the cell, and here's one of them. Here's a receptor protein, which is waiting for this particular steroid hormone to come along. So the steroid hormone comes along, it interacts with the receptor protein. They make a complex, which is called a receptor hormone complex. In turn, then, these move together directly into the nucleus, where they can interact directly with DNA. Isn't that powerful and amazing? Now, it takes a little while for this entire system to get going, but once it does get going, it is incredibly powerful. So usually steroid hormones will have a little bit of a lag period, like more of a lag period. It could be hours for this signal to really get going, but once it does get going, it's very powerful, and it oftentimes lasts a long time. So the steroid hormones then, this receptor hormone complex, moves right into the nucleus and binds to your DNA, and look what happens next. It's going to bind to a particular gene, and it's going to say it's going to upregulate that gene. So it's going to manufacture a bunch of mRNA. That mRNA transcript then is able to leave directly out of the nucleus and interact with ribosomes. There they are, 1, 2, 3. This particular mRNA transcript can actually make hundreds of new proteins. So this is a ribosome that is spitting out a new protein, but you'll notice there's one right behind it spitting out a little bit more, one right behind that that's getting ready to spit out some protein. So one particular, again, this level of sensitivity here is amazing. You just have one hormone here interacting with one receptor, making one receptor hormone complex. They get inside of the nucleus, they start interacting with the DNA, and the longer they sit in here, the more mRNA transcripts that they make, And the more mRNA transcripts that they make, the more protein that they're able to manufacture. So you can see the time, you can see why it is that the time course is a little bit longer here. It takes a while for all of that transcription and translation to occur, but once it does occur, you have tons of new product to work with. This cell has been changed. It's taken a while for it to change, but it's been changed profoundly and for some time.